So now it's my pleasure to introduce the next presentation, which is focused on the Flexter project. So Flexter is one of the integrated projects Yassine introduced this morning. So a quite efficient tool to collaborate within the LabX network with complementary expertise we have in the consortium. And I think you will see it's really a perfect example of this with a very wide range of topics and results we will see in the next minutes. So the presentation will be made by Yassine, Haddab, and Philippe Cinquin. So Yassine is full professor at University of Montpellier. He's head of the Dexter team, working on design, modeling, and control of robots for manipulation. And Yassine is also the recent co-founder of a company, AQ Surgical, and he has a research expertise on the design and control of microsystems, medical surgical devices. Philippe is director of Team CIMAG, famous lab in Grenoble, co-director of CICIT in Grenoble as well. And he's actually one of the pioneers of CAMI and also the first coordinator of our LabX. He's I would say, a serial entrepreneur, in addition to be a researcher in the field of biocells, computer-assisted surgery. And today they will share with us the results of the Flexter project, from new knowledge on GI microbiota to a company creation. So Philippe and Yassine, the floor is yours.
Thank you very much. Thank you very much, Pierre, for the introduction. And good afternoon to all. So I'm Yassine and from Lyon, and I have the great pleasure to share this presentation with Professor Philippe Cinquin. Our presentation today will be divided into 4 parts. The first one aims to describe the context in which the work was carried out. Parts 2 and 3 present 2 projects for the development of sampling capsules for intestinal microbiota. The first had led to the creation of a startup, and the second has explored bio-robotic interactions using bistable mechanical structures and 3D printing. The last part will be devoted to the conclusion and future work. So let's start with the introduction. So the talk today is based on the work done within the framework of the Flexter integrated project that I will present in the next slides, and also on the previous work carried out at TIMC on implantable glucose biofuel cell and symbiotic microbiota modulator. On this slide, you can see the contributors of this work from the 3 labs, and from Lyon, eCube, and TIMC, and some of them were funded by CAMI LabX. Some words now on the Flexter integrated project. Flexter stands for Flexible Medical Microrobotics for Gastrointestinal Diagnosis and Therapy. It's a close collaboration between 3 laboratories in CAMI, Lyon, TIMC, and eCube. So the project objectives are the development, design, and fabrication of microrobotic systems for non-invasive medical tasks, diagnosis and therapies linked to the GIT, the gastrointestinal tract environment, with the link with well-known diseases. Design of new concepts of microbiorobots transiting through the GIT or staying inside for long-term therapies, and the exploration of patient-friendly and environment-friendly operation principles derived from micro-nanotechnologies, additive manufacturing, and biology. So this work was made possible by sharing the multidisciplinary skills of the 3 laboratories in the field of miniaturization system design, additive manufacturing, mechanical simulation and optimization, biology, and experimental development. Several projects have been developed in Flexter, but we have chosen to focus our talk today on the design of capsules for intestinal microbiota sampling. So, sorry, okay. So capsules have been used for several decades for drug delivery. They are designed to deliver their contents to the inside of the intestine without being damaged by the harsh conditions in the stomach. The first technology capsules were designed to perform imaging functions. They pass through the gastrointestinal tract by the same principle as drug capsules, using peristaltic movements. So later, many capsules were designed and produced to perform various other functions in the gastrointestinal tract. You can see some examples here. They often consist of a large number of precision mechanical parts. They are sometimes difficult to control and use bulky external devices for their operation. Our goal has been to develop a new generation of safe, patient-friendly, and environment-friendly capsules. So the main features of our designs are non-invasive sampling of the intestine microbiota based on ingestible capsules, no embedded electronics, no batteries, no complicated mechanics, but the use of biorobotics interactions for navigation and sampling actuation. Taking a sample of the intestinal microbiota using a simple capsule requires several tasks. The capsule must be easily swallowed, protected during its passage through the stomach. It must take the sample and protect it until it leaves the digestive tract. So solutions must be found for each of these tasks. And in the following, in the two projects, we will show how these logs have been lifted. So we will switch now to the description of the first project.
Thank you, Yassine. Thank you, Pierre, for the introduction. So following the introduction of Yassine, I will describe how this was implemented. The principle that was described by Yassine was implemented, as you can see here, with a first capsule, which is not so big. We will see later how big it is, and in which you have several individual capsules which are capable of performing the sampling. So the idea is that the first relatively big capsule is gastro-resistant, so it passes through the stomach, and then it dissolves when it arrives in the duodenum. So each of here the 5 independent sampling capsules become free. As you can see, the principle is that each of these capsules is made of an elastomer, and inside you have a sort of spring, which is made of a foam that we will describe later, and this is blocked by a plug. And the control of the device comes from the characteristics of this plug, because this plug has the capacity to dissolve, and depending on the choice that you do, the duration of dissolving will be shorter or longer. So each of the plug will dissolve at a time, and if you have programmed it correctly, one will open in the duodenum, the other in the jejunum, and the final one, for instance, in the ileum. So you have here the possibility with a single capsule to examine, to analyze the characteristics of the complete in a small intestine. Next, please, Yassine. So the foam that we are using is PVA, which is polyvinyl alcohol. And in fact, the idea is that this PVA is both the energy source and the motor for sampling. But before we see how it works, you have to know that it's completely biocompatible. It's even a dietary ingredient. We have been told that it is, for instance, in the Coca-Cola. I don't know if it's true. And then it's also a polymer which you can find either as an hydrogel or as a foam, and we are using the foam. And it's a super absorbing polymer, as you can see here. And it's used in various medical devices. Next, please. So you see here the way the device is working, because it is capable, when you pour water on it, to grow bigger, and you see that as a result, this implies a force, and you see on the graph the way this force can be computed, and it can be significant. In this instance, it can go up to 3 newtons, for instance. So when it takes the water, it releases the energy that has been embarked by compression, which has been stored by the compression, and in turn it absorbs water, and in the water, of course, there are the bacteria that we are interested in. And then the bacteria may live, because as you see in the microscopic image at the bottom, you have a porous structure, which is very favorable for the life of the bacteria. Next, please. Well, you have here the successive prototypes. The very first one dates back to 2016. And we first started with very simple mechanism, which were nitrile gloves. And we designed with this first prototype, in which we had put the PVA foam. Then we had a prototype which was more adequate, in which we were already using a silicone. We also have been exploring the possibility to have 3D printing. And now we are using molded and extruded silicone. So the constraints that we have to take into account are constraints about the size, the sampling volume, the sealing is very important, and we have also to take into account regulatory issues, because this has to be used by human beings. And finally, you have to design something which can be massively produced. So today, in a single 0,0 capsule, so as you see on the right, it's a little more than 2 centimeters in length and 7 millimeters in diameter. We can put 3 of our capsules. Each of them is 150 microliters, completely biocompatible. And as you will see in the slides to come, perfectly sealed when swollen, and it's adequate for the small series production. And what you can see on the bottom at the right are images of the capsule once it has swollen. So you see in the middle, I don't have the mouse, but I guess you can see the blue part, which is a single, thank you, Yassine, which is a single capsule which has swollen. On the right part, you can see 3 of these capsules which are embarked in a single 0,0 capsule, which you can see as a transparent. And on the left part of this panel, you have the X-ray image of this the device, because it's important that you can track it if something goes wrong. Of course, there is no reason why it should go wrong, but risk analysis implies that you take this into account. Next, please. So what you can see here is how it works in terms of capacity of sealing. So we have performed lead tests, and first you have to see which is the level of pressure that we have to undergo. When you contract your anus, the maximal pressure is about 300 millimeters of mercury, and this corresponds to about 40,000 Newton per square meter. And if you take into account the complete surface of the capsule, and you take half of it, because when you compress it, you touch half of the surface, you see that it amounts to 82 square millimeter, which corresponds for a 300 millimeter of pressure to 3.3 newtons. So on the left part, you see a device that was not working adequately, because you are able to see on the video, and you see on the left part that there is some leaking. No, it's the left one, yeah. The left one, yeah. And you can see that it has leaked at some point because the sealing was not perfect. And yet the force was only 1 Newton. So this version was not adequate. We modified it. And now with a force of 10 Newton, which is 3 times what is necessary, as you can see here, when you compress, there is nothing which happens. The leak is 0, so the device is completely sealed. Next, please.
Okay.
So how does this work in real life? Well, of course, you have to retrieve the device, the capsules. So there is a device which is called a fecal cleaner, in which your stools have to be stored, and then it can be folded and sent by courier to the laboratory. So you get the device. The silicone is blue. So with a knife, you open it. In the middle, you see that you have opened with your knife. So you see the blue part, which is the silicone coat, and the white part, which is the foam of PVA. And in the end, at the right part, you have isolated the PVA sample. So now you put the PVA sample into a container which is adequate for centrifugation, which is represented below. And on the right part at the bottom, you see that after centrifugation, the liquid has been expelled out of the foam and is ready for analysis. Next, please. So for the proof of concept, we first worked on in vitro characterization. So we analyzed the characteristics of the bacteria which were in the middle in which we had been working and inside the device, and we were able to prove that they were very similar. And then we went to animal trial. We used a pig which swallowed the capsule. And then we retrieved the capsule and we performed a bacterial analysis, both by culture of the bacteria, direct culture, and also by metagenomics, in which you characterize the various types of the bacteria. And with this, we were able to prove that indeed the capsule had opened at the right place, and that it was possible to characterize the content which corresponded to the intestinal content expected in a pig. Next, please. Well, now we are at the stage of a clinical trial. The promoter will be Grenoble Hospital, Sugar. The principal investigator is our colleague, Professor Nicolas Mathieu, who is a gastroenterologist, specialist of this type of pathologies. And the main objective of this clinical study is to demonstrate the safety of the device and to demonstrate that it goes well through the digestive tract. So the idea for this proof of concept is a short, limited number of healthy volunteers, 15 volunteers. And we also have a secondary objective, which is to demonstrate that indeed what we retrieve is not fecal matter, but really the matter, the liquid of the intestine at the level of the small intestine. And this will be proven by metabolomic analysis on one hand, and on the other hand, by metagenomic analysis with shotgun 60S sequencing. So our target is proximal jejunum and ileum. And we were waiting for the final versions, which, as you saw, is now ready. So time to start it. It will be ready probably by the beginning of second quarter of 2021. Next, please. Well, these were the good conditions to create the startup company. And I have a pleasure to announce you that it was created less than 1 month ago, because it was November the 26th. And the CEO is Thomas Soranzo. who, by the way, is a previous CAMI Flexter postdoctoral student. And so, of course, we are very happy to have him as the CEO of this company. And you can see the first product of this company, in which you see the gastro-resistant capsule open at the bottom. You see the 3 capsules which will be put inside this capsule, and then you see it closed, and finally you see it in the device, in the wrapping, as it will be sent to the patient. Next, please. And that's for you, Yassine. Thank you.
Okay. Okay, thank you very much, Philippe. I will now speak about the second project, which is the design of a 3D printed sampling capsule based on bistable mechanism. So in this second project developed during the PhD thesis of Muna Ben Salem, the requirements for interactions with the gastrointestinal tract are identical to those presented by Philippe. In this case, we have exploited interactions inspired by robotic interactions, robotics at high precision mechanics. We allow as maximum dimensions those of the largest pharmaceutical capsule, the 000. The dimensions are given here, so 9 millimeters in diameter and 26 millimeter in length. So the goal here is to perform sampling also in the intestine, and to avoid this area around the stomach where the pH is close to 1 or 2, which is very, very low. And then after that, the capsule can go to the small intestine, where the level of the pH go up to 5 to 7, and here the capsule can be can perform the sampling, and after that the capsule is retrieved. So the scenario is the same as previously described. However, here we try to use an approach using a bistable structure. So here you can see the different parts of the capsule. So it consists of 2 parts shell that you can see on the left here, a bistable mechanism, and a PVA foam that you can see here. So it works on the same principle as described previously. So let's see now the operating mode of this sampling capsule. From the mouth to the stomach, the capsule is protected by the coating, and the mechanical bistable remains in its first stable states. So the first stable states is what you see here, this first shape. Okay, so during the first step, the capsule is protected by the coating. So once in the small intestine, the coating dissolves, then the intestinal liquid enters inside the capsule. So the PVA foam here absorbs the liquid and triggers the bistable structure. So here the sponge is also, the foam is also used both for sampling of the microbiota of the intestinal liquid, and also as an actuator to switch, to trigger the bistable. For the rest of the path, the capsule is closed and sealed, and the sample is protected until the recovery. So what's bistable structure? In this video, you can see how a mechanical bistable works. It has 2 stable states. The application of a force allows to switch from one state to the other, and no energy is required to keep it on one of its stable states. On the left, you can see the basic shape of bistaple structure used in this project. So the design of the capsule components required significant developments in miniaturization, mechanical studies, simulation, and experimentation. For example, the design must respect many constraints: small dimensions, enough force, and also different forces between the two stable states. The bistable must be triggered by a low force produced here by the PVA form, and must have a much higher closing force here to avoid the contamination of the sample. This is achieved by high precision design of this part of the capsule. Here you can see the force displacement characteristics, and this symmetry allow us to obtain this effect. So it's not so easy to fabricate, to manufacture this system in very small dimensions. So we have used 3D printing for manufacture of this capsule, and we had to solve some problems for, to obtain the good mechanical characteristics. And we know that mechanical properties, for example, of materials are very different in the fabricated parts, and they differ from the raw material, for example. And the manufacturing depends on the orientation of the parts, because there is anisotropy of mechanical properties. And it is sometimes difficult to control the trajectory of the nozzle. So if you don't take care of the design, you may have this kind of problems where there is a fragile link here. So we had to modify all these designs to obtain the right characteristics. And this design, we have verified the operating of this design. Here you can see the video showing the operating of the system. Here you can see the capsule inside liquid, and now the water, the liquid, is entering by this hole. The PVA foam is expanding inside the capsule, and it is applying a force on the bistable beam, the bistable structure. And when the force reaches a certain amount, it triggers the structure. That you see here that the gas, the air bubble is going out. And here we reach the force level required to trigger the bistable, and you will see in a few seconds okay, so here you can see the switching of this device. Okay, so okay. So to complete the design of this system, we have also designed here a silicone seal that was added in order to ensure that the capsule is watertight. So the sampling and sealing have been successfully tested in vitro in an artificial liquid similar to intestinal liquid. Here you can find the characteristics on the left of commercially available liquid that imitate the characteristics of the intestine environment. Okay, so the characteristics of the sample was the design and the test have been performed in vitro for this design. Okay, so for the last part, please, Philippe, can you?
Yeah.
Okay.
vous m'entendez pas là?
Yes, we do hear you.
Okay, sorry. I was trying to find the distance control. I'm sorry. Yeah. So as a conclusion, I think that it's important to remind you that we are targeting a new way of performing medicine, in the sense that the imbalance of intestinal microbiota has a major impact on probably all chronic diseases. On the left part, you have some instance of diseases with, for instance, inflammatory diseases of the small intestine, or neurodegenerative pathologies, diabetes, neurological disorders such as depression and cardiac failure, for instance. And so this relationship between microbiota disorders and this pathology is proven by a growing number of publications. It's exponentially growing, about twice in each 5 years more publication. About 27% of the clinical trials are devoted to this type of pathologies. And if you look at the markets, it's about €2 billion a year. So, of course, having the possibility to have an impact on this is very important on the various domains, such as nutrition, such as also diagnosis, such as therapy, or also monitoring of therapy. So this is our target. Next, please. In order to be able to analyze the content of the intestine, the present solutions are not adequate because their limits are too important. Of course, you can work on animals, but then this raises ethical issues. Mostly, it's very difficult to transpose your results to human beings. There may be risks, and also it's expensive. I would say 95% of what we know about the microbiota comes from fecal sampling. But the major limitation is that it's not at all representative of what you have in your small intestine, and this is where many interesting things occur. Finally, you can do through endoscopy. This is feasible, but this is very aggressive, very invasive. It implies sedation of the patient, so it cannot be done in real life, for instance. It has to be done only in the hospital, and it's very expensive. So really, if you discuss with any physician specialist of chronic disease, probably they will say you that it's very important to get access to a method which gives real access and faithful access to the intestinal reality. And this has to be implemented easily at a large scale. This is a requirement from the clinicians. Next, please. Well, this is exactly where we are today. I think that what we have shown in this Flexter project is that, yes, it is possible to fulfill these requirements and to combine the data that will come from this multi-omic analysis of the intestinal data with other data, which are the clinical data and the other biological data. So it's a typical issue for artificial intelligence because you have to merge all these data and to extract what is important for you. This means that you will have to perform clinical trials, statistical analysis to characterize the heterogeneity of the intestine in various pathologies. And of course, once you have identified these new biomarkers, you come with new patient and the sample of his or her intestine will give access to a new biomarker for the pathology. So this is why we call it intestomics, because it means that we combine all sorts of multi-omic information about the patient. Next, please. And as a summary, what we have shown in this presentation is that we are able to access the intestinal content in a simple, non-invasive, affordable, and scalable way. And that these signatures can be used for prognosis, diagnosis, therapy monitoring, and also for the development of new therapy and nutrition solutions, thanks to an analysis based on artificial intelligence of all these data, which are very heterogeneous. So what we hope is that we enter a new type of approach of these pathologies. Again, this corresponds to basically all chronic pathologies. Next, please. And this is the end of this talk with the list of publications that were made both in papers, journals, and conferences, and also patents. And by the way, you can see that we went very quickly because the first patent dates back to 2016, and 4 years after, the startup company is created. Thank you. Thanks for your attention.
Thank you very much, Philippe. Thank you very much, Yassine, for this great presentation with many, many perspectives. So we have time for a limited number of questions to stay on time. I could read one from Jérôme. Jérôme, do you want to ask yourself? Or I can do it. The question was about the maturity of the pills. So especially the blue components. What's the TRL of those solutions today? How mature is it from a technological point of view?
Well, it's very close to the higher levels of TRL, so probably it would be 8 or 9. I don't know exactly. Meaning that we have a device which is operational, which is ready for clinical trial on healthy volunteers, and which will undergo CE marking on the basis of this clinical trial.
Right, thank you. Another question maybe on the principle from Matthias, who is asking about the fact that once the external membrane is dissolved, then you have several independent capsules, or do they stay together? So about the principle.
Yeah, it's important that they are independent. In fact, each of them lives its own life. Well, there are reasons to think that they will stay close because they are in the same flow, but it's not mandatory. And anyway, each of them is independent. And remember that what we plan is to develop adequate plugs so that each of them is programmed to open at a specific part of the small intestine. So each of the capsule is independent, and there is no reason why they should stay close together.
All right. There is also a question to Yassine about the bistable mechanism, and if you have some basic numbers or an idea of the force needed to exclude the air from the device when the water goes in. So do you have some elements?
In fact, it depends on the design of the PVA foam. Currently, maybe Muna, you can give the last version of the capsule if you are hearing me. No, she's not here. Okay.
Yes.
Okay. Okay. Can you please, Muna, give the last values used for the triggering of the bistable structure inside the capsule?
Mm-hmm. So the trigger of the bistable mechanism fabricated with 3D printing. We did characterization phase for this bistable mechanism.
Muna, we are not hearing you very well. If you.
Oh, sorry. Do you hear now?
Yes, more or less. Yes. So if you just have some value about the maximum force, the force you can sustain with the mechanism.
Yeah, so the maximum force needed to trigger the bistable mechanism and close the capsule is around 0.35 Newton. And once the capsule is closed, the mechanism is able to resist to up to 2.5 Newton in the sorry.
Okay, so 0.35 to close and 2.5, that is the force that can be sustained by the mechanism. Okay,
yeah, exactly.
Thank you, Muna. All right. So I think we have no more questions. So thanks again to our speakers.